exciting to see some new research in this new environment and wonderful facilities. So the title of my talk today is talking about how our immune system or innate immune responses contribute to axonal degeneration in motor neuron disease. And this particular form of inflammation or inflammatory pathways we are interested in particularly is called type 1 interference. So for the audience today, I think I'm not going to too much about MND or disease pathology in too much details. But all we know is the progressive neurodegenerative disease pretty much hitting the motor neurons, which is in our in the brain and spinal cord, control our movement and control us how to breathe and talk. So as many of you know, the 90% of these disease cases are sporadic, and only 10% are familial. So that means only a few patients or cases are related to their family histories. But in general, like other neurodegenerative disorders, the major risk factors contribute to this pathology is including aging, smoking, infection, and most of the environmental insult that in general increase the stress in our environment and therefore impact on our cell homeostasis. Of course, based on the genetic database, we know there are some genes they play a causative roles or even risk factors. They contribute to MND development. And this including the major genes including SOP1, TAR-DBP, which is the gene encoding the TDP-43 protein we're going to talk about today, FAST, C9orf72, TBK1, and so on. And the emerging studies showing actually now many of ALS patients, they don't really only carry one genes, but multiple. So that is already opened a new avenue into a new insight to study whether MND is just a group of, is a one disease or is a group of diseases. And that's why also explaining how disease might develop or contribute to its heterogeneity. So talking about that, right? No matter what kind of conditions they contribute to MND or causing MND, almost 97%, almost all patients with MND, or 97%, but based on literatures, the patient of MND associated with the cytoplasmic TDP inclusion pathology, or in short, people just call it TDP pathology. And it's just the postmortem samples from different patients in the spinal cord and also motor cortex, showing different morphology of TDP inclusion stainings. So you can see some are fibril, some are pig body-like, and some just around the nucleus. So that again indicating, the different progressions of different presentation or manifestations in these patients. But not just MND, this pathology also been shown in other conditions like frontotemporal dementia patients. So not just MND again, I think this TDP pathology is a disease hallmark across the ALS FTD disease spectrum. So when we talk about TDP-43, it's a very well-known DNA RNA binding protein in the nucleus as shown in green here in neurons. And then so that regulates RNA processing, including splicing, transportation in and out of nucleus, and stabilize the mRNA for protein homeostasis using its two RNA recognition motifs, 1 and 2, flanked by nuclear localized signals and export signals. But very interesting, it has a very interesting C-terminal glycine-rich domain. This is the prion features. So by itself, upon stress or, for example, infection, or even by toxins or any injury inside cells, this protein, because the structure of this C-terminus already prone to misfolded and mislocated into the cytosol and aggregate by itself. And this is also a very interesting domains where most of the ALS causing mutations located today. So what is the major difference between mutant TDP compared to wild-type TDP, given they all have this glycine-rich domain? Based on the literatures and also using this in vitro model as example, when you overexpress express wild-type TDP-43, it can cause TDP pathology by forming this puncta around the neuroid in these neurons. But mutant can further push this in a more aggressive way. As you can see, more puncta, and then even causing more damage to neuroid, as shown in these IF images. So for a long time, people using overexpression models to study gain of toxicity functions of TDP-43, led mimicking what happened inside of patients. But it's not always the case. And I would like to, before my talk, I would like to share a very interesting theory proposed by Associate Professor Meg FightMND from the University of Zurich, who you may already know from this network. So she actually in Basel this year in the International MND Symposium, and showing there are major two pathomechanisms by TDP pathology they drive to LS or MND in neurons. So first is what we just talked about due to the gain of toxicity, and that is due to TDP mislocalized, accumulate and recruiting more TDP protein, and they cause neurotoxicity and propagation pathways. So that's why people working in these fields, they try to identify how or decide the agents they can target these pathological forms of TDP and blocking it from aggregation, and therefore you can restore its function in neurons. But the other party of the scientists working on loss of function, because when TDP mislocalize and stay outside of nucleus, it will lose its intrinsic DNA RNA processing mechanism. So that is called loss of function. So they are associated with misregulation of RNA targets, and this including a lot of great research published in Nature recently about the loss of STAMIN2, ANK13, and increased MPTX2, which all play an important role to maintain motor neuron development and survival. So in this case, people try to target the downstream RNA defect mechanism to reverse its toxicity caused by loss of function TDP. But what she really proposed is this might be ongoing in the real-time fashion. So for example, TDP might mislocalize into the cytosol first, that cause the early acute gain of toxicity function followed by loss of function in the nucleus. So I think this is really interesting, to the field. When she first time present in Basel in the International Symposium MND Research, because we don't have to debating which part mechanism is to contribute to or driving to MND. It can be happen in the real-time fashion. So I would like to talk about type I interferon today, and this is building on what is followed by this ongoing TDP pathology with gain of toxicity or loss of function, because from different RNA-seq data and also proteomics data recently, for example, from Adam Walker's lab, showing there's an increased type I interferon signature in the mouse brain or cell models. But also when people using knockout to delete TDP-43, they also see increased type I interferon signature. So what is type I interferon signature? I think maybe across all your research, you might see from your qPCR, from your RNA-seq, or from your protein analysis, you might see any of these targets pop up in your candidate list. So who are them? Actually, type I interferon is the antiviral signaling as a part of defense mechanism in our immune system. So it's very well known, maybe based on what we learned so far from influenza or COVID infections. Interferon is to shut down the protein replication process inside of cells. So how they work? So when we get infected, and then your cell get infected with virus, for example, they will try to replicate inside of your cell by producing a lot of particles of virus for their genome as well. So this including a lot of viral proteins, and also viral DNA or RNA. So this will activate a range of immune receptors, and then they generate cytokines called type I interferons. So type I interferons including interferon alpha, interferon beta. You don't need much, you just need a tiny bit. They are able to bind to the interferon alpha beta receptor They are heterodimer. It's formed by subtype 1 and 2, and 1 is the dominant role. So that's why scientists always delete infnut 1 in mouse models or in cell models to study its pathways, and which we're going to use later in our studies. So when they bounce to these infnut receptors, they will activate JAK1 start pathways for transcription of interferon stimulate genes. So all the genes he named here or protein, they are all the family under interferon stimulate gene or ISGs. So mainly what they're doing is the beauty of immune responses. They will try to shut down the protein stress, and then by producing more chaperone protein, cope with in order to cope with the unfolded protein responses, because you have a lot of protein generated inside of cells. But also they want to increase more immune sensing and signaling. Then finally, I think they want to get the friend to help to the site by recruiting the helper to the site of infection. So that's why you see chemokines like CXCL9, CXCL10, and CCL2. Sorry. So why this pathway antiviral is so promising inside of in terms of neuron degeneration? Because when you think about, you have a TDP-43 pathology or any form of protein aggregations, let's somehow mimicking viral infection inside of our cell. In this case, neuron can do it as well. So, and also there are a list of drugs already approved by FDA. They're showing they are safe. They are potent to shut down this pathway. For example, baricitinib, tofacitinib, ruxolitinib. These already been proved for using rheumatoid arthritis and also for viral infection, including COVID severe cases. So the question is, can we target type 1 interferon signaling for LS? So while we're talking about this, actually, there's a clinical trial currently recruiting patients from Boston, within 2 hospitals in Boston. They want to test the JAK1 inhibitors, baricitinib, to treat 80 patients LS and also asymptomatic carrier of C9 of patients or carriers, using 2 doses up to 6 months treatment, and in order to see if they can shut down inflammatory biomarkers in their CSF, including some key cytokines. They are associated with type 1 interference, and also the neuron degenerative markers like a neurofilament light chain. But at the moment, they're just recruiting patients. But most important thing is I felt very interesting is they don't know the mechanism how interference driving LS or driving neuron degeneration or neuronal loss. But now they are already recruiting patients to do this. And given baricitinib is not really brain, they don't have a very strong data from supporting its brain penetrant or not. So I'm not too sure whether these can really reach the brain for effective outcomes. But I think, who knows? They are, although we don't have a strong data for showing that, there are some preclinical data showing baricitinib can somehow get into the brain. So let's cross fingers for that. So back to our work. So interferon signaling can that be a new therapeutic target for LS? Actually, it's not new. So back to 10 years ago, when people use SOAP1 as a LS models, they showed us an increased interferon signaling in astrocyte from SOAP1 mutant mice and also in microglia. And that's how they believe the glial cells that activate by interference become neurotoxic or reactive to neurons' health. But again, they just crossed to infnut knockout to delete the signaling. They didn't show why and how interferon drive neurodegeneration in molecular events, or we don't know the mechanisms. So as many of you know that we show that TDP pathology or stress can trigger mitochondrial DNA release that activate the cGAS-STIM pathways, generating cytokines, including type 1 interference. Therefore, that will downstream hitting the infnut and interference signaling in the paracrine or autocrine manner. But again, this is just one of the explanation for genotoxicity theory. And the cool thing is we're not just showing in glial cells. In this study, we're showing in human iPSC motor neuron and also patient spinal cord postmortem samples. So again, this study just provide one angle of TDP pathology driving this, somehow leading to interference, but maybe there's also loss of function when TDP accumulate outside of the nucleus. So in general, we still don't know whether type of interference can really contribute to LS development. So to address that questions, we use a very well-known TDP mutant mouse models that carry human transgenic ASV15T mutations under the prion promoter. So that increases overexpression in the brain and spinal cord with the flag tag. So we can trace whether they carry this transgenic protein. And then since I moved to Florey, actually we restudied and rephenotyped this mouse model in more details. For example, my postdoc Samana repeat the IF imaging to show this mouse actually have a very nice TDP pathology. As you can see in green, for the human form of TDP that mislocalized with the beta-3 in red in neurons, and this is in cortical layer 5 of the brain. And some they are not just mislocalized in the neuron, but also there are some red negative, green positive cells. We've seen that's also happening in glial cells. So we know this mouse model have a very severe gut blockage issues, but we now managed to resolve that issues and also supported by many publications these days. So by giving them a diet gel or gelified food, they can resolve and making to avoid the gut blockage or fund hardening. So that will buy us another 2 to 3 months of the time frame to see a very nice progressive neuron degeneration. For example, these mice will be totally fine before 90 days of age. And on 90 days, that's the onset. We were using the published scoring systems to quantify their gaze development or dark field development. So then that will generate these progressive motor deficit. As you can see in the videos, this will develop these motor phenotypes over the time from 90 days to 150 days until they cannot upright themselves up to 30 seconds. And that is mark the end point of the experiment for survival, but not literally they die from this phenotype. Yep. So the first thing is seeing is believing. So when you grabbing these mice, when we cross to if not heterozygous knockout or homozygous knockout, you see that's very clearly after onset about day 120, if not knockout to mutant mice, they're showing some kind of protection. However, heterozygous doesn't show that. They still have a very severe motor phenotype with this gait, a severe gait phenotype compared to control. And this the loss of infant actually slowing down the time taken to develop to hitting the end points of experiment by 24%, which is shown in green. So, for example, in mutant, you can see the median date of the survival is 145 to 150, but now they can extend it to 170 roughly. But still, kind of not as good as what we showed before when we crossed to STING knockout, and that's because STING is more upstream of interference. So probably that's why infant knockout only protecting half, because STING can also activate the other inflammatory pathway called NF-κB. But strikingly, that's still providing significantly slowing down or mitigation of disease progression, which is evidenced by the rotor performance in green. Again, we can see the trend of if not heterozygous knockout to mutant, but it's not significant, but it's a nice trend, which we will show in the in other studies. But homozygous doing very good job at the age of day 120. In terms of neuron degeneration or neuronal loss, we use nasal body staining or quasar blue staining to quantify overall neuron count. So as you see in the layer cortical layer 5, which is more severe hitting by in these models, infant knockout in green actually slowing down the neuronal loss. And then although it's still less than control, but that shows a very nice protection. So this is just brain. How about spinal cord? Because that's where TDP also overexpressed. So we can see again from Samana's IF, you can see they also have a very profounding TDP mislocalization, and then that increased the type of interferon gene expression by qPCR, interferon beta, and MX1, IP1, IP44, and then of course crossing to infant knockout, or when they lost the receptors, they don't express these cytokines anymore. And then in terms of neurodegeneration in their ventral horns, and that is very profound as well. You can see it's slowing down the progression and all protect them very well, at least at this stage. So now we see interference increase in the brain and spinal cord. So that will, we know from the antiviral signaling, we understand that will do something to the brain. So again, that's not always a reason why cells express cytokines. And then, so whether they are doing shutdown protein protein production or try to generate chaperone for refolding protein aggregation, something like that, that is an interesting thing. So one thing we want to see is the neuron inflammatory event. So one hallmark is the inflammatory monocyte infiltration from peripheral blood into the brain. And this is the example done by Professor Steiner lab from the Northwestern University in US, showing very nice human motor cortex in healthy. There's only a slight a very little expression of MCP1, which is monocyte chemotactis protein 1 in magenta. But in LS patients with TDP pathology, you see they express a lot that are responsible to recruiting monocyte into the brain via this CCL2 CCR to MCP1 CCR2 signaling axis. And very interesting to see in the star symbols here, you can see they are colocalized with the M2, which neuron can express these chemokines, not just the microglia, what we know for a long time, because we think they are resident immune cells in the brain, so they should primarily express cytokines or inflammation, but actually neuron can do it from their data. So to prove this concept, actually we use single cell flow cytometry to process our H315T mouse brain. And then we use the standard immunology getting strategy to purify immune cell from local microglia. And then from these CD45 positive cells, most of them are neutrophils and monocyte. So again, in H315T mice in blue, we can see there's an increased immune cells. Most of them are monocyte, but also neutrophil, but that are not shown. But this infiltration of immune cell from peripheral can be fully prevented when we cross to infant knockout. Suggests this is a secondary effect, but what happened inside the brain, that is more primary causes. So to address that, we're using IHC to standing for IBA1, which is the marker for microglia. You can see there's an increased microglia in the cortical layer 5, although they are highly expressed throughout the brain. But when we cross to infant knockout, you see the number of IBA1 positive cells is reduced to control levels. In this case, we're only interested in layer 5 because that's where LS hits in these mouse models. Strikingly, I want to highlight is the GFAP staining, which is the astrocyte. So you see a really nice accumulation of GFAP positive cells in this cortical layer 5. Now it can be prevented very well when we cross to infant knockouts at this age. of mice, 120. And not just the cortical layer 5, we also see this accumulation between the GFAP positive cells between cortex and also hippocampus. And I think, please correct me if I'm wrong, from literatures or a mouse autonomy, this region is the white matter. And then they're showing, the literature suggests increased white matter GFAP accumulation, indicating axonal degeneration or damage and aging as well. So this is a very good sign that we shut down this certain inflammatory pathway. We can sort of, addressing the possibility of the reason of axonal degeneration in these mouse brains. But altogether, these inflammatory monocyte recruitment and also GFAP, IBA1 gliosis, they can be just a paraconmanant or secondary effect. The question we want to ask next is which cell type or who is the troublemaker in this brain by producing type 1 interference. So of course, we're using RNA in situ hybridization or RNA-scope to quantify cells with ITGEM, which is CD11B for all myeloid cells, including monocyte and microglia in the brain. And we see about only 35% of these cells express type 1 interferon in response to TDP overexpression. Importantly, do the similar thing for ROBS-FOX3, which is expressing neuron for all neuronal markers. So we see about 60%. So although we see 60 versus 35% of cells expressing type 1 interference, but maybe myeloid cells as an intrinsic immune cell type, they are more responsive. So although they have a less percentage of cell expression, but maybe intensity of cytokine expression can be much higher compared to neurons. But this tells us the neurons can also express type 1 interferon, which is very nicely consistent with the previous finding in CCL2 data. So we know neuron can produce type 1 interference, and they also express interferon receptors. So is there a possibility that neuron can trigger neuron toxicity or neuron degenerative cascade by itself through this cell autonomous fashion? To address that, we're using sushi cells, and so it's the neuroblastoma cell line. Then we use the DOX-inducible flag-tag TDP-43 with wild type and Q321K. And then after induction of TDP using DOX, we give them a baricitinib or tofacitinib. Here, data only show for baricitinib. So by Western blots, we see just like what we published earlier in C-Gastin papers. We know this construct in neurons can induce phospho-TBK1, which is downstream of C-Gastin pathways. And then they also have phospho-STAR1, which is the paracrine interferon signal. Baricitinib selectively only shut down type 1 interferon, but not the primary TBK1 downstream of STIM. And then we can see the treatment shut down the type 1 interferons and ISG's transcriptions by qPCR. More strikingly is these increased type 1 interferon actually have a more LDH release, indicating the cells are stressed, damaged with their ruptured membranes. Not necessarily dying, because LDH assay is not can be quantified when cell is stressed, not just dying. But with the baricitinib treatment in the dose curve, you can see they reduce these cell neuron toxicity or cell stress in dose-dependent manner. But again, this is just a cell line. They're still proliferating, and also they don't have a proper neuronal morphology, for example, axons. So to address that, we use iPSCs from 3 healthy patients, 3 healthy controls and 3 TDP mutations. And then we use the dual protocol to get the mature MX1 double positive motor neurons, and then we give them the baricitinib for the final 28 days. Again, you see these cells, as published before, they have an increased type 1 interferon, MX1, and also IFID44, and then can be inhibited very well by baricitinib. But most beautiful thing is when we do IF at the last stage and endpoint of these iPSC neurons, one of the LS, this is just showing one representative image from LS patients. You see these axons in white are damaged in this DOT discontinuous morphology, while cell bodies still remain intact. And baricitinib can somehow protect that very well. So this is the quantification showing cross 3 LS mutant TDP mutant lines can be protected very well by treatment of baricitinib. So what happened to these axons? Actually, we don't know. So that spent us pretty much 2 years to find a potential mechanism for that. And then we found a very two interesting observation. One is when we increase interference, and then we can see using sushi cells as a model, we see the mitochondria, which is standing in yellow. They're supposed to be around the nucleus, but now they kind of unpacked and they start extending to the end of the axons. Of course, sushi doesn't have a very beautiful axon, but you can see they're moving to the end of terminals or the edge of the cell boundary. And with increase, with the dose of interferon increase, this is more profound. And of course, cell is going to react to these disruptions. So what we see is there's an increased modification called polyglutamation. So that is the modification by adding more glutamate peptide onto the beta tubulins, indicating the tubulin out of the cells, especially neurons, they are under stress. So that making the all axonal transport, especially the retrograde, very sticky to transport things, cargos back, and maybe that's why we see mitochondria stay outside of the nucleus into the axons. But this is all very good sign from the literature showing this is the very strong characteristics of axonal degeneration at the early event. So what is the mechanism for this? Because when we go back to iPSC and neurons, we see this morphology pretty much just hitting the axons, but not the cell bodies, as they all remain intact at this stage. And then from our typical view of cell death, they were associated with nucleus or mitochondrial fragmentation, but we didn't see that here in these cells. So that remind us the programmed axonal death pathways, or in other terms, Wallerian degeneration pathways. So how they work actually, so this is mediated by a protein called steroid alpha motif 1 or SUM1 protein. SUM1 normally remain inactive when cell has a high level of NAD+. So many of you working in neuroscience, you know that is the supplement for mitochondria, maintain neuronal, keeps neuron happy. So when our cells under trauma, damage, stress, you have a less NAD+ remain in the cells. They will force someone to undergo a conformational change. They will extending their TLR domains, which is the NAD+ hydrolase. So they will consume more remaining NAD+, and they will trigger cell death. So this Steinbeck phenotype or phenomenon actually will start from the terminal, but if we don't remove the damage or the stress didn't get resolved, this will progress towards the soma of the cell and then hitting mitochondria or nucleus, and then that will cause the cell death at the end. So again, someone is not very new in LS because the early study from the SOA mice, they're showing they are only little or no protection in SOA mice, although they see very nice neural degeneration biomarker reduced when they cross SOA1 to SUM1 knockout mice. But there's no protection in terms of survival. But given SOA1 can be more aggressive and they already have an early soma and mitochondrial damage compared to TDP models, so their mechanisms or maybe this different mechanism might due to might causing or contribute to this difference in disease phenotype. But still, again, we don't know the role of someone in SOD1 in human conditions. So they remain to be studied and to be investigated. And then, but earlier study also showing someone knockout instead have a protection to stop neuron from degeneration, and also to prevent the neuromuscular junction denervation in another TDP models carry Q331K. So is this someone also only specific to TDP? That will be very interesting, and then we are addressing that question. But again, to highlight the role of someone in LS, I think that's very clear by the recent study that identified the hyperreactive SOD1 mutations in the hydrolase motif that cause LS. So I think, again, it's like how aggressive these mutations by TDP or SOD1, or whether how someone is activated in the axonal terminal ends or near the cell body in the soma, they were also leading to different outcome in the mouse models. And again, Eli Lilly not just having a baricitinib, but also they now purchase these disarm inhibitors to shut down SOD1 activities. And then now they want to do this in a clinical trial for a couple of neurodegenerative disorders, including ALS. So finally, I would like to quickly talk about some of preliminary data we have. So at the end, we want to know, we see interferon is important, and then we also see someone can be activated. So in LS, so are they linked together or just parallel? So to use the for the proof of concept results, we use sushi cells, and then we give them a interferon beta response dose curve to mimic what happened downstream of interferon TDP pathology in neurons. So phospho-STAT1 showing very nice in interferon activation pathways. And key thing is we see there's a dose-dependent SOD1 expression. This might indicating there's an increased activity, but we don't know at this stage. But also very interesting, we see when we increase interferon or inflammation in these neurons, they also have phospho-TDP-43. So whether or not interference or infection or stress, they can induce TDP pathology. That's another angle, but it's not in this. We have other project to study that, but not in this project. But that's a very interesting finding. In mouse models or X315T, cortex layer 5, someone showing very strong green tails in these axons colocalized with beta-3 tubulin, and also they accumulate in the cytosol or soma near the nucleus. So showing, yeah, they do, someone do actually increase or elevated in X315T mice. So how to quantify its activity, not just study expression. So normally we know upon insult, our neurons lost the NAD plus, and then people normally using a standard mass spectrometry to quantify this ratio of NMN and NAD plus ratio to indicate SOD1 activation. But now we are with our collaborators, Dr. Andre Lorito from Sydney, and they now have a very nice tool called PC6, which is the paradigm analog to mimic NAD+. So that will be utilized by active SOD1 when it's activated, generating PUP6, which will be GFP signals. So using Vacro as a SOD1 activator as a positive control in these primary mouse neurons, Andre showed very nice PC6 activation, and also there are the stimulation triggers for someone in the much mild and slower motion can be also detected by this PC6 dye. So we are in the process now to using PC6 to quantify its GFP signals in our iPSC neurons carrying TDP mutations. So finally, be over time now. So I would like to summarize. We found the type 1 interferon inflammation pathways trigger axonal degeneration in TDP-43 protein-apophyse models. So when we delete inflammatory knockouts in the ACE1 mice in vivo, we see prevented inflammatory immune cell infiltrations, protect them from microgliosis and astrogliosis, and then this leading to less neuronal loss in the cortical layer 5, and of course, a better performance by rotarod. A novelty is showing that neuron in the brain of TDP mice can also express an interferon beta, not just, and then this suggests it's no more a feature, only feature for immune cells in the brain like microglia. And interferon expressions correlate with cell autonomous neurotoxicity. And then we see in sushi cells, we found mitochondria get stuck in the axonal or the cell terminal end with sort of interesting finding in polyglutamation. And also we see increased SOM1 expression and perhaps TDP pathology. In vivo of ACE1 mouse, we also see that increase only in neuron. That is very striking. And then we found that a profound axonal cell body accumulation upon disease onset. So given someone has a double role in axonal degeneration, as we just discussed, but they also play a role to induce more inflammatory responses like phagocytosis and also cytokines. So whether or not this, our study showing type 1 interferon links to someone activity, they might contribute to propagation of the whole disease into LS development. So what we are doing at the moment is we are going to test this PC6 dye to quantify activity of someone in our iPSC motor neuron from patients, and then would be interesting to see what's the impact when we're using baricitinib with or without the disarm inhibitors. Now it's already available and both owned by Eli Lilly as a combination treatment to these neurons. And because this already been shown for several conferences, we decided to finish this manuscript, and then we are going to submit to bioRxiv by July this year. And of course, in terms of treatment, I think we get approached by a discovery from Cambridge. They want to use AAV microRNA someone to achieve last and consistent specific reduction of someone in the brain of ACE1 mice only under one single injection. And that would be very interesting to continue this collaboration with the industry. And finally, I would like to acknowledge everyone involved in my in this work and collaborators and funding bodies. Thank you.
Fantastic. Thank you so much, Alan, for your presentation. Heaps and heaps of data and a really exciting topic. For anyone who doesn't know me, I'm Bekshi, and I'm the Director of Research and Programs at FightMND, and I'll be looking after our question time today. Just a reminder, if you do have a question, to please use the raise your hand tool, or if you're having trouble doing that, you can pop your question in the chat. Alan, I can see we've got a really great number of people attended today, so I'm sure we're going to have some great questions. But while people are working out how to raise their hand and pop the questions in the chat, I might jump in with the first question. And my question's about, so we know from the literature that throughout a disease progression, both in animal models and in patients as well, that the role of inflammation changes. So just wondering about interferon 1 and whether there's temporal changes as the disease progresses, and what that would mean for treatment, as far as, you know, a blanket sort of knockout or knockdown might not work when we're looking at the right timing of those types of treatments.
That's a very interesting question. And then I think, in general, as an immunologist, we always believe cytokine release, you know, although people say it's when they link inflammation, when they think about inflammation, they always think about chronic responses, but actually not. So inflammation actually in the is try to they trigger in the acute rapid pathways. So the idea is they just try to light the fire and get the attendance, and then let the helpers do their job. So yes. So for targeting information, a certain information or inflammatory pathways, we need to know which pathway drive this. And then second, I think normally it's happen in the early stage. So from the RNA-seq and also proteomics studies, you only can see a very nice information or markers they increase at the early stage. Only at the early stage. So yes, we need to identify the pathways, and the beauty of innate immune pathways is you not just getting the kinase or the targets for therapeutic or inflammatory or for the inhibitor to bind to, but also you can identify a messengers, metabolite, and also the downstream factors as a biomarker for that. So we need to identify a right biomarker to detect a certain or specific inflammatory pathways to allow us to find the right timing to shut it down. Yeah.
Great, thanks, Alan. Any questions from the floor? Great, we've got a question
Nice.
from Mary Louise. Okay, Mary Louise, you should be able to ask your question now.
Yeah. Great talk, Alan. Thank you.
Oh, thanks.
I was just wondering about, the changes that you see early on in the. Do you think that's similar to the changes in Adam's mouse? Because that's early on. So your changes and the changes with overexpression, do they give the same profile?
That's very interesting question too, because so Adam did that RNLS8 mouse models for proteomics, and there's no follow-up proteomics for A315T at the moment. And given, I think it would be slightly different, because that's the Adam's mouse is the wild type TDP, and then it's very it's kind of very like accelerating models. They get really sick when they're off the dog's feet between 4 to 6 weeks. But for A315T mice, that takes about the whole lifespan is up to 150 days. So, and given the time span may be different, and also the different form of mutant or wild type TDP, it can be hitting something very different in between. But having said that, it can be something in common as well. So I think we need to do a proteomics in this A315T. Maybe not just us, but anyone using or interested in these models in Australia. I think we can find a way to do this proteomics. But before that, I can't really say much about whether or not they are very alike or not. But based on TDP pathology, we can probably find what's in common. For example, they all have interference responses, but we cannot compare at the moment without proteomics data.
Okay, thank you.
Sorry, yeah.
No, thank you.
Thanks, Mary Louise. We've got another question from Sophia Lukinga. If we could just introduce ourselves and where we're from before we ask our question, that'd be great. Thank you.
Hi, Alan. Hi, I'm Sophia. I'm from the Florey as well.
Yes, I know you, Sophia.
Hey, well, thank you for your talk. I was just wondering, you say knocking out or like reducing interferon can improve the phenotype. Do you think that increased interferon also can drive TDP pathology?
In neuroblastoma cell line sushi, we showed that, and that's very interesting, but we don't know yet, because first, that phospho-TDP we see in cell lines is the soluble lies in repark fraction. So that is kind of conflicting with what we know today. We always think the TDP get phosphorylated and become like an insoluble aggregate. So we're still in process to prove that. But there are some studies from immunology papers showing when they inject interference into the wild type mice, they do generate, they induce aging or senescence phenotype, but also they might have some motor death effect too. And this is also been shown in LS models, in Alzheimer's model, when they're injecting interferon into the APP mouse brains, they also have a severe neuronal loss in hippocampus. So I think it's very likely that will happen in vivo too.
Great. Thank you, Alan. We've got another question from Andrew Toslini in the chat. Hi, Alan. Thank you for a really nice talk. Is there any evidence of SARM1 involvement in spinal cord motor neurons or at the NMJ?
Yes, I think, although I didn't show the data, but we do have the spinal cord data from the brain, someone's IF staining. And then they also show the similar thing. So it's very likely.
Great.
Yeah.
Thanks, Alan. Thanks for the question, Andrew. And I had another question for you, Alan. Just around, so do you think that the role that interferon 1 is having is purely a Wallerian degeneration? Is that purely driving disease through that process?
Not really. So I think we discussed that in this year's international symposium on MND. And so I only showed that pretty much all based on neuronal data, but someone from Cardiff also showed interferon actually required for microglia. So I think it's very likely this is cell type specific. And then we need to study more in terms of, first, of course, by collaboration. We are not able to study everything in our hands. But second, I think we can do something like a conditional knockout, only knocking out interference inside of neurons. And then given interferon can also, I think, it's a cytokine for inflammation, but when we talk about when it's just released in the very early time point, maybe it's beneficial because, for example, for microglia, it's important to activate them by inflammation for uptake, phagocytosis, the TDP aggregation, or when it's propagate between cells. But what happened after, the early events, it can be somehow change microglia or astrocyte phenotype towards more damaging, and then therefore neuron, which is already very vulnerable or susceptible to this inflammation, they're just getting worse. But again, what I want to say is the cell type specific.
Great. Thanks, Alan. Well, we're running out of time, so I will throw back to Sarah.
Thanks so much, Beck, for taking the question time today, and thank you so much, Alan, for joining us today.
Thank you, my pleasure.
Yeah, it's really interesting to see some of the exciting results you have out of your laboratory, and really looking forward to seeing some of the next steps of this project and where it can take you. If there's anything else that anyone would like to follow up with Dr. Yu today, you can get in touch directly with him or with us here at FightMND if you would like us to help connect you. The seminar has been recorded today, so if you do know of anyone who wasn't able to attend live but would like to catch up, you can let them know that it will be available on our website in a few days' time. Well, thank you everyone again for joining us today for the FightMND April Scientific Seminar. We hope you've enjoyed today's presentation. Please join us again next month when we'll have a new speaker, and keep a lookout in your emails for news on this seminar, which will take place on Tuesday, May 28th. Well, thank you, and we'll see you next time. Thanks, Alan.